All right, welcome back. I hope you're having an amazing time so far. Our next speaker is Abhishek Pandey. He is Group Lead Pharma Discovery at Abbvie. Please join me in welcoming him to our virtual stage.
Hello, I'm just trying to switch on my camera. Okay, yeah, it's on. Let me share my screen. Great. So today I'll be going to speak about a topic which is very new and it's less explored, let's say it that way, multiomics and machine learning in pharma. And I'll try to discuss some of the use cases in pharma where people can use it or people are trying to use it. And all the data is actually from the reference in terms of literature. So there is all of these things are published in some way or form. So there is a basis to it of all of these use cases that I'm talking about. So when I say multiomics, actually, in many ways and forms, multiomics can be of multiple different meaning. For example, for single cell people, multiomics means just combining proteomics with metabolomics, with lots of other omics data together. That could be a multiomics as well. And then for people like me who are on the machine learning side, multiomics means combining any modalities together, any two different modalities together. Hopefully they are orthogonal. By orthogonal, I mean that they are complementary to each other. That'll be even better. So that's why we try to use multiomics analysis. And one of the key factors why multiomics analysis should be useful is actually imagine that you have a lot of different modes of data. You have different types of omics data that is available, and you want to actually give a insight for all of these data together rather than actually just using single mode of data. For example, just the genomics analysis or just the imaging analysis or just the pathology analysis or just the proteomics analysis. So rather than doing just one mode of analysis and building insights and predictions out of just one mode of data, what about if we combine all different modes of data together? What about if we combine the power of genomics with imaging? And when I say imaging, it could be pathology or radiology. What if we can combine the power of imaging plus genomics plus clinical data plus some other mode of data that is available out there that we haven't yet explored? I think that is the most powerful tool that could exist out there, because you are trying to use all the data that is available for patient. You are just not deciding on one single mode of data. You are trying to actually decide on the whole mode of data. And how I normally say it is that when you use multiomics analysis, you are trying to actually understand the whole elephant. So for example, radiology images could be considered as elephant. When you look at the tissues of elephant, that's pathology. When you look at the genomics of the elephant, that's essentially trying to understand what genomics elephant is made up of. But at the end of the day, all of them are complementary information. And to actually complete the picture of disease in an elephant, you need the whole picture. You need the elephant, you need the tissue slides, you need the genomics, you need all of them together to actually fulfill the complete picture for the disease and for the patient. So that's why I say that multi-omics analysis could be something that we should be all doing. And we should be actually thinking about it, if we are not doing it actually. So then comes the question of machine learning, and to that I always come up to these slides, which very beautifully actually explains this. So for example, the cost of producing a new drug is actually increasing day by day, and it's like $2.6 billion per drug to bring to market. And cost is one thing, but then comes the time. It takes around 10 to 15 years to actually produce that drug and bring it to market. All of this, so bringing the drug to market is costly as well as time-taking process. So how do we do something, or how do we change something within this process that can actually reduce the cost as well as time? And one of the greatest example of it will be computation. We should be bringing more and more computation. We should be bringing more and more machine learning to our methodologies and whatever we do in our labs, so that we can actually reduce the time of discovery for a drug and bringing it to the market. And if you see on the right side, there is only this 3 to 6 year cycle is what you can actually affect with your machine learning. Most of the time, the 6 to 7 year clinical trial, FDA review, and rest of it we have no power over it, because clinical trial will run at its own speed, FDA approval will go at its own speed. But one thing that we can control is actually the drug discovery and the preclinical portion. So this 3 to 6 year period of like discovery and preclinical analysis, I think we can reduce, and we want to actually discover some of those drugs which will be cheaper, because we'll be employing algorithms to do it rather than doing hit-and trials in labs. And the other thing is we should be actually doing more sort of like analysis based on computation. That will be faster also. So that's why we'll be able to reduce the time. So I think switching more of our lab assays to lab analysis that we do and takes time, switching them to machine learning would be a very neat idea to go forward with it. Switching back to multiomics then. So how do all of this fit? So I'll discuss today 4 different use cases for multiomics analysis. And all of these 4 different cases, as I said, are currently all focused towards pharma world, how pharmaceutical industries can use it. But as I said, it's not limited just to pharma. Many of these things could be used in multiple different places and in multiple different healthcare paradigms. So one of the first things that I would discuss today is the target identification. So for the pharma world, actually, most of the people or most of the big pharma companies have good enough medicinal chemists and people who can actually design molecule and drug the undruggable, if that's the word that if you can say those words. But the problem is that most of the time we do not still know a proper target. And most of the time how we search for our target is by going through the literature, years and years of literature, and just trying to understand if there are any indications for a target in the literature. And many times we try to do some sort of like genomics analysis where that kind of signal can also come out. So target identification or trying to understand or find a new target for a disease is one of the most primary thing that pharma industry would be interested in. And we can use multiomics analysis to actually come up with targets actually. And that's actually one of the cool things that have been developed recent years. Of course, all of this will be used with machine learning, but as I said, machine learning will be a tool to actually employ some of these things, but multiomics is the crux of all of them actually. Then comes the preclinical disease understanding and pathways. We actually, as I said earlier, the preclinical stage is also something that we should be actually affecting. And whatever we develop in the preclinical stage, whatever kind of multiomics insight we build in the preclinical stage, hopefully if we can translate it to the clinical side, I think that's half the battle won, if you may call it. Because what you're doing then is that your drug is actually characterized, it has been pointed out, and then your drug actually can be understood properly in the preclinical stage, and hopefully the same understanding can go further down the line. And then comes the clinical insight. The clinical insight is essentially when you actually have done you are on the clinical side of things, we have crossed the preclinical stage. You can still use multiomics analysis and the clinical side of things, and then we can move towards precision medicine in the clinical side of things using the multiomics analysis. So the first of them is the target identification. So the target identification using deep phenotyping in large human cohort was done by Calico. This is the paper that Calico came up with. Calico is the life sciences arm of the Google. And what they did is that, for example, they use the UK Biobank data. And for example, if you're trying to follow a pancreatic disease or a liver disease, essentially they segment out, of course, they use deep learning to segment out some of these organs. So they segment out the organ from the MRI scans that were acquired in UK Biobank data. And these patients were followed across time, because if you understand UK Biobank data, it's voluntary a lot of patients were actually scanned, as well as genomics data was acquired for them, clinical data was acquired for them across time. So as they were followed across time, for example, in pancreas or liver, the first thing they did is they come up with a signal or a signature, imaging-based signature for these patients. And once these imaging-based signatures were extracted, then what you can do is you can relate or correlate these imaging-based signatures with the GWAS-PWAS kind of study and try to say that what were the genes which were responsible. For example, if there is a liver disease and the signature comes out to be the increase in liver volume or increase in the texture of liver, things like that, then essentially that would correspond to a liver disease, and then correlating it with a GWAS-PWAS study to come up with a genomics marker, essentially saying that, okay, these are the genes which are responsible for this liver disease, and essentially then verifying it with lots of statistical analysis for the proper signal, and then saying that these are the essentially the signals that actually would be considered as targets. And that's how there is a method to come up with target. Of course, this is one way of applying the data. Of course, there are multiple other ways of using the multiomics analysis to find out target, but definitely this is one of the best ways that I have come across, at least in the literature, that actually can show that we can come up with targets in different therapeutic areas. And of course, Calico's whole goal is in aging. We can actually do it in other places as well. Then comes the biomarker, multiomics biomarker development and the workflow in the preclinical space. So what we can do is in the preclinical spaces, for example, we can have our images, whichever images we may have. These are mouse kidneys that we are looking at. And then you can segment out the kidney, again, using the machine learning, deep learning algorithm. And then once the algorithm segments out the kidney, then you can extract features out of it. And then those imaging features can be combined with the RNA-seq data, with the preclinical data, or the biomarker data, and then come up with like a multi-omics insight for the preclinical stage. And hopefully some of those insights, if done properly, can translate to the other stage, which is the clinical stage. And that could be like as I said earlier, it will be a big deal if we can do that. So there are multiple ways you can use actually multi-omics analysis. One of the most standard ways is actually you just go through the data and you essentially just combine them together. So concatenating different modes of data together, let's put it that way. When you concatenate different modes of data together, for example, RNA-seq or MRI data or preclinical data or proteomics data or metabolomics data, you can come up with an insight which can actually be more powerful than the single modes of data, as depicted in this mock-up table, where the C index improves when you combine the clinical, genomics, and imaging data together, and the integration has better C index than the rest of them. But just from my experience, actually, this story may not be exactly true, but there are different modes that you can combine together. Definitely, that will be way better than single mode, and that I can actually give from my experience. But there are other multiple ways you can use multiomics analysis. Rather than combining all of the data or all of the features at the very beginning of it, what we can do is we can actually have more sequential flow for multiomics analysis. For example, use imaging to stratify your patients into a progressor and non-progressor group. And then if you have RNA-seq data, what you can do is you can actually look at the pathways that a progressor group adopted versus what the pathways that a non-progressor group adopted. So that way you can actually come up with the pathway along with the biomarker to stratify the patients, and then say that these are the pathway that a progressor adopted, and that you can use it in your studies down the line. So here are some of the examples of exactly two different workflows that are published in literature. And of course, I have given the link to the exact papers as well. So essentially, the left one is as the left one is in here, where you combine different modes of data together. You concatenate them together before you do the prediction or insight. That's essentially the left paper where they combine the imaging plus genomics plus clinical data together to come up with better survival curves for oncology patients in lung cancer. And one on the right side is essentially the sequential way that I was telling, where you actually use imaging to actually stratify the patients and then use RNA-seq data to come up with pathways that actually the progressor or the good performing patients adopted versus non-progressor. And there are more examples of both of them actually in literature, but I just put 2 examples here. And then comes machine learning portion. And I have to end it very fast here. Essentially, machine learning can help integrate the data in different ways. Of course, as I said, in the imaging portion, machine learning could be used to segment out the data and things like that, but it can be used to actually integrate the data in its own way. And this is one of the paper where you do an orthogonal fusion of data of radiology, histology, sequencing, all of them together in a deep learning sense, and come up with actually better predictions. And if you see here, the fusion data set have way better predictions than actually single modes of data. And they have way better C indexes than single mode of data. So that's the improvement I'm talking about whenever I'm saying that the multiomics can bring that kind of change to our analysis. And with that, I think I'm running out of time, so I'll end there. And thank you for listening to me. And I hope to answer your questions in the next session.
Amazing, Abhishek. Thank you so much for sharing with us today. This was an incredible presentation, and I know this virtual audience is giving you a huge virtual round of applause. Thank you again. And for the audience, it's time for you to make your way to your next session. Along the way, make sure you accept your connection request and take some time to check out our awesome exhibits. Thanks so much, and we'll see you around.